Good morning. I'm Gul. It's now my pleasure to introduce Dr. Philip Lee, co-founder and chief technology officer of Senti Bio. The format for this session is a roughly 25-minute fireside chat. And if you have any questions during the session, feel free to type them into the question box and we can try to ask them on your behalf. So thanks again for joining us, Philip. Senti is moving towards its first IND filing this year, so it's an exciting time. To start off, can you give us an overview of Senti and its manufacturing capabilities before we dive into some details?
Sure. Thank you, Gula, and happy to walk through some of the key advances that we've developed on the manufacturing side. So Senti Bio, as many of you may know, is working on advanced cell and gene therapies. So the core of what Senti Bio has developed over the past handful of years is really centered on our gene circuit technologies. So these are methods where we can program genetic functions into different cell and gene therapies. And these, some of the examples are listed here, including multi-arming, logic gating, regulator dial, as well as smart sensors. And we've demonstrated this across a number of modalities, such as NK cells, T cells, in vivo gene therapies, as well as stem cells. We are working on a couple of collaborations on the in vivo gene therapies and iPSCs. And internally, our pipeline is focused on allogeneic NK cells in that top box. And that's largely what I'll talk about in terms of our manufacturing capabilities. So as we've been developing these allogeneic cell therapy products, we realized very early on that having process analytical development capabilities as well as GMP manufacturing capabilities really needed to keep pace with the advances in the science and the product development aspect. In order to do that, we have established two key facilities, both in the Bay Area. On the lower right there, we have a research development and technical operations facility in South San Francisco, allowing us to look at optimization across numerous modalities, viral vectors, as well as cell therapies, and really think about how we optimize in a product-specific context. And then we've also recently built out and have up and running a GMP manufacturing facility. There's a picture of that on the upper right. It's a 92,000 square foot building, also in the Bay Area in Alameda, California, where we have a clean room set up for clinical manufacturing of our NK products. So just as a quick overview of our lead program, which we call Senti 202, this is a allogeneic CAR NK product, which is targeted for AML and other blood cancers. So if you look at that schematic on the left side, in blue is a healthy NK cell, which is what we're starting with. And we have engineered these cells to express a number of components, which will really enhance their capability against difficult to treat cancers such as AML. On that top panel, we have a bivalent activating CAR, which targets CD33 and FLT3 to get deeper clearance of the leukemic cells. On the right side is a inhibitory CAR, so to protect healthy cells. In this particular case, primarily to protect healthy bone marrow cells. And then on the bottom, we express a calibrated release IL-15 cytokine, which enhances persistence and durability of the NK cells. And this program is, as Gula mentioned, on track for an IND this year, and we're looking forward to getting this product into the clinic. On the manufacturing side, early on, as I mentioned earlier, because we can leverage our gene circuit technology across multiple cell types, including different sources of NK cells, and we did evaluate different ways to make Senti 202. We have decided to focus on peripheral blood-derived NK cells, so on the right side of this slide. I won't go into all the detail here, but some of the key reasons for that selection were the ability to maintain that full repertoire of functional and mature NK cells that are found in healthy adults. And we've also developed a manufacturing process which is very scalable with this source. And then we can also take advantage of a mature supply chain and regulatory understanding of peripheral blood cells, which have been used in multiple clinical trials to date. So our current manufacturing process is outlined in this slide. At a high level, as I mentioned earlier, we are starting with healthy donor blood. So this allows us to obtain material from multiple individuals. The NK cells are isolated and they're stored frozen. So this gives us the further capability to test the cells prior to making the actual clinical doses. Step 2, we are using a viral vector currently to do the cell engineering of the NK cells. And then in step 3, they're expanded many thousands of fold to get a very large batch to have multiple doses per batch. These cells are then vialed into standard drug vials and frozen so they could be stored and delivered off the shelf to patients. And then finally, just to wrap up the overview, where we are today with manufacturing, we have a process which is very amenable to the current cell therapy supply chain. This gives us rapid access and scalability in terms of starting materials with the donor NK cells, using our optimized viral vector for the genetic engineering, as well as supporting materials such as engineered feeder cells to help with the activation and expansion of the NK cells. We do have a manufacturing facility up and running currently in the Bay Area, and this is supporting our lead program as well as additional activities that we were looking to get into the clinic. And then we do believe that this paradigm of an allogeneic CAR NK cell empowered by our gene circuits really gives us the potential to both scale up our process as we progress and also broaden patient access, because NK cells, as well as the allogeneic nature, allow us to have a safe product that can be infused across multiple patients. So we are continuing to work on next generation technology development to further scale our process, as well as to improve our manufacturing capabilities. So I'll pause there with the overview and happy to address any additional questions to talk more about our technologies.
Great. So you touched on this a little bit, but what do you see as the most differentiated elements in Senti's manufacturing process from those of other cell therapies or even other NK cell programs specifically? For example, are there proprietary elements that could give Senti's products an edge?
Great. Yeah, a lot of the core differentiation really comes from our gene circuit technologies, and we spent many years working through hundreds, if not thousands, of different designs of each of our products. So in the Senti 202 example, a lot of where we see the potential benefit is in how we've engineered that construct. So for example, the NOT gate in 202 is something that we think is very differentiated. What that allows, if you think about a typical CAR T or CAR NK product, you can target and kill cells that express a receptor. But unless you have a very clean antigen that's only expressed on cancer cells and not expressed in any other tissues in the body, you are limited in that therapeutic window. So having this NOT gate allows us to both target cancer cells more aggressively, as well as protect healthy cells such as the hematopoietic stem cells. And so that we think is very interesting. In addition, for NK cells, our calibrated release IL-15 is something that's unique. IL-15 has been shown clinically to be very important for NK cells to enhance their activity as well as to maintain persistence. But with the calibrated release IL-15 in a single construct, what we can do there is to both have an autocrine effect, because it is localized to the surrounding space of the NK cells, as well as because it is released into the microenvironment that can trigger additional endogenous immune system response. And this is something that can be done with either membrane-bound or secreted IL-15. Secondly, I think on the manufacturing side, what we found over the years is, for these gene circuit constructs, there is a lot of optimization that's necessary in order to maximize your properties. For example, if you think about the genetic engineering we're doing, it's a fairly large construct that we're putting into these NK cells. So how we optimize that sequence, how we design the viral vectors, how we perform the transduction process, as well as the expansion, all of these are very important to optimize for each product type. And then the final piece, which we can dive into a little bit deeper later, is because we are starting with healthy donor cells, there is a lot of work in terms of understanding the properties of healthy donors, which are best suited for each of our product types. So that includes the donor selection aspect, as well as how we harvest, cryopreserve, and store the products for off-the-shelf use.
Yeah, so I just want to follow up on a couple of points that you just talked about. So in terms of the viral vector that you use, first of all, do you use a single viral vector to put in, all of the components? And secondly, do you make that viral vector in-house also?
Right. So for the Senti 202 product, a lot of the work I mentioned earlier was to find ways to package that entire transcript into a single viral vector. As some people may know, NK cells inherently are more finicky in terms of not being very happy when they're virally modified. So compared to other cells like T cells and stem cells. So a lot of the work we did was, how do we optimize that viral vector to both package the fairly large construct that we're putting in there with the multiple gene circuit elements, as well as to maintain that cell health and performance of the NK cells post-transduction. So we currently do use a single retroviral vector product for that, which is great. It's very efficient. We can get very high expression levels from the NK cells, and we don't see any reduction in our expansion potential. In terms of the manufacturing, because we have worked through internally through all of the technical operations aspect of that, and we have a existing GMP facility, we are set up to do in-house viral manufacturing. That being said, there are external manufacturing partners which have capabilities that we can leverage as well for this material. So we do think there's a lot of innovation in terms of how we design the vector, but the manufacturing process is something that could be done both in-house as well as externally.
Mm-hmm. Got it. And then you mentioned the parameters associated with donor-derived cells and kind of how that might impact the NK cell product. Can you expand a little bit more about the kind of work that you're doing on that front to characterize donor screening and other elements there regarding consistency and potency for your manufacturing process?
Sure. Yeah, the field of allogeneic NK cells is, I'd say very exciting right now. There's initial clinical data that's very promising in terms of safety when dosing with allogeneic cells, so you don't have the graft versus host concerns that you do have with T cells, for example. So I think from that perspective, there is a very good foundation in terms of allogeneic use of NK cells. The additional scientific question that raises, which honestly I think the field is still trying to better understand, is, are there differences between donors that will lead to better clinical effects? I think there's a lot of data being generated. The jury's still out in terms of how to define that. But I think from our perspective, we've set up the process where we can obtain from leukapheresis material from healthy adult donors, very high purity, good quality NK cells. So that's the first step in our manufacturing process, which is done in our GMP facility. That material is actually vialed and frozen. So what that also gives us the ability to do is, once we collect starting material from multiple donors, we can characterize vials from those banks to understand how they would perform in our process. So currently we do this, I would say, semi-empirically, in the sense of looking at in a smaller scale, a transduction with our viral vector and gene circuits, looking at those expandability between donors, as well as the functional properties of those transduced NK cells. And in that work, we are able to see that certain donors do have better performance than others, and those can be pre-selected to use in the actual clinical product. In terms of the actual markers to look for that predict improved behavior, that's a data set we are generating. And I think we're fairly encouraged with what we're seeing so far, that both from a practical aspect, because of the way the process is set up, and then also from a scientific aspect, as we're understanding some of these correlations, that these will be useful as we go to larger patient numbers in the future.
So on the topic of going to larger patient numbers, the slides that you presented talk a little bit about expansion of the NK cells. Can you talk a little bit about the scale that you're at, and to what extent you expect that to be sufficient for a commercial launch, and how do what kind of levers do you have to further scale?
Right. So from a process perspective, our initial targets, which we started developing a couple of years ago, essentially is to get to that 100 doses per batch level. That's definitely a good sweet spot in terms of clinical development. From a commercial perspective, we are aiming to go higher, but maybe I'll start on the first part of, if you think about existing autologous cell therapies, you have essentially one batch or one dose per batch or one patient per batch. And so the benefit of an allogeneic product is you can greatly improve that, in theory, closer to what you would see in a traditional biologic setting where a single batch could serve many, many patients. So in terms of getting to that 100 doses per batch number, what that typically means in an NK process is for a single batch, if you can get expansion of the NK cells in a few thousandfold, so if you're starting with tens of millions of cells, if you expand them a few thousand folds, you can get them to, call it 100 billion plus cells per batch. That tends to be a good sweet spot in terms of clinical development, which is what we're focused on now. Our expandability, which we've seen, and partly this is benefiting from selecting key donors, but also the way we do the biotransduction and also the activation and expansion of the cells tends to be in the, we've seen batches averaging about 6,000 fold. We've seen, 10,000 fold in others. So we do think from a scaling perspective, we are in the right ballpark. Thinking about the commercial scale aspect, one of the interesting things with NK cells is, ourselves as well as others have been able to take these cells and expand them fairly far in the sense that you can get them to keep dividing. There are still some questions in terms of what's the trade-off in performance clinically that has not been fully tested. But in our hands, for example, we've been able to expand NK cells using certain approaches, at least 100,000 fold, close to 1 million fold as well. So I think from a biology perspective, it's doable. It would involve larger bioreactors, sort of a slightly different process equipment in order to handle a 10-fold increase. You're talking about maybe 1,000 vials per batch. But those are capabilities that do exist in the field in terms of, you think about other biologic products, other ways that cell therapies can be made. But realistically, in today's market, it's really just limited by that demand question. But we do feel pretty confident that as our products move through development, we will have capable technologies to continue that scaling.
And so obviously at this point, we don't know what exactly the final doses will be for NK cell product, but it is looking like NK cells will have kind of a multi-dose paradigm as opposed to the one-time dose treatment for CAR T cells that we've seen. So how do you factor that into your manufacturing in terms of your estimates of batches and things like that?
Yeah, that's a very interesting part of the NK field today is, given the safety profile, and in many of these trials, there is a matching across the donor and the patient. And I believe there's also data on you could actually have different donors being dosed to the same patient as well. So I think that gives us a lot of flexibility in terms of our manufacturing paradigm, being very much more like a traditional manufacturing approach. The key thing I think we think about from a technology perspective is similar to the previous question. If we can get maximal number of doses per batch from a single donor preparation, that gives us a lot of capability to use that across different patients. The other thing I didn't mention earlier is in the way we're collecting the NK cells, a single donor collection can actually serve many batches of material. So even if a single batch is 100 doses, a single donor collection could be many-fold that. And then you can also get material from that same donor multiple times. So I think that gives us, with a starting pool of different donors, I think a lot of different options in terms of how we do our batch manufacturing to serve the broader patient population. And definitely as we get into that scale, I think there'll be some really interesting questions of, can we find even better clinical correlations of how certain patients respond to material from different donors. But I think that's all to be determined as more data comes in. But from a manufacturing perspective, we're pretty happy with the flexibility that this type of paradigm gives us.
Great. And so at a high level, can you orient us a little bit to where your process is relative to autologous cell therapies in terms of cost, or in another way, could you talk about kind of the major cost drivers for your process?
Sure. Yeah, I think, just to start off, like an autologous T cell process is definitely more mature, I would say, from a manufacturing perspective. There are commercial products. There's been a lot of experience in terms of end-to-end, how to manufacture and commercialize those. So, at least to compare where we are today with the allogeneic NK cells, which largely are in, phase 1/2 trials across the board. I think there's a couple of key differences which we anticipate would help from a cost perspective. One, the most obvious being an allogeneic donor-derived material is not constrained by the patient-specific aspect. So the cost of that manufacturing batch can be spread across multiple doses, very similar to a biologic product. So what that ratio is, still kind of TBD in terms of how NK cells scale. But as I mentioned earlier, it should be in the ballpark of 100 to 1 to 1,000 to 1 in terms of that math of just dividing across patient doses. In our experience, the cost and the underlying process of doing a batch is relatively similar, whether you're making a single dose or a single patient or 10 or 100. So I think that's really where you're going to see the cost benefits in terms of, as we get to these larger scales, the cost per dose should come down. Just as an example, if the day before harvest, if you're able to double the cells, one extra doubling, that essentially translates into 50% less cost for your end product, because that additional day of expansion costs relatively little compared to the overall cost of that batch. So I think those are the things that we will continue to push on, thinking about the ultimate cost per dose of these therapies.
Great. So as you mentioned, the NK cell space is still emerging in terms of clinical development. But can you talk a little bit about the analytic methods and approaches that you use to characterize what you're looking for in terms of the product? Again, appreciating that less is known about what you might be looking for relative to the CAR T space.
Yeah, I think one of the fascinating things, at least scientifically, that we found as we develop products like Senti 202 is that it gives us a lot of ability to think about the analytical aspect. And particularly with these gene circuits, if you think about them, they're engineered programs. We've designed these cells to perform a certain function. So the fun thing on the analytical aspect is, how do we then characterize those functions? I'd say largely because we have specific characteristics that we're looking for. So for example, the activating CARs and the inhibitory CAR activity, we can design very interesting and specific assays that can read out in terms of that function across different constructs, across different batches. So I think that is a very interesting area where there's a lot of technical development. In relation to that, I think from a regulatory perspective, there's been a lot of good guidance recently in terms of how companies like Senti should think about CAR cell therapy development. What are those properties that are important to look at? How do we make sure that from a product-specific perspective, we can demonstrate that our design product is performing the functions that we expect it to? Those things come together very well in terms of the analytical strategy. And I feel like the field in general is moving forward, very, very quickly in that space.
So just in the last minute or so, obviously, the current financing climate has been challenging across the space. Any comments on how that has impacted your manufacturing priorities with respect to your programs?
Yeah, I think our underlying strategy remains the same. We do see a lot of value in being able to quickly move advanced gene circuit products into the clinic to get that clinical proof of concept. Our internal capabilities really help us do that. I think we've seen tremendous progress even in the last few months in terms of how we can move, these somewhat complex products quickly into this GMP process to enable an IND as well as initial clinical data.
Fantastic. So I see we're at the end of our session time. So I'd like to thank you again, Philip, for your participation and for the great overview.
Great, thank you.